The overall goal of these staining methods is to quantify intracellular lipid abundance and evaluate lipid distribution among different tissues.
This method can help answer key questions in lipid metabolism, such as how are lipids genetically and biochemically regulated, and which disease states dysregulate lipid homeostasis.
The main advantage of this technique is that it is relatively simple and inexpensive to carry out.
To carry out Nile red lipid staining, plate worms on nematode growth medium, or NGM, seeded with late log OP50 E. coli, and grow the worms at 20 degrees Celsius to early L4 stage. Wash the worms with 1 milliliter of PBST solution and transfer the worm suspension to a 1.5 milliliter microfuge tube. Centrifuge the worms at 560 times gravity for 1 minute. Then remove the supernatant and repeat the PBST wash until the E. coli is cleared from the suspension. Next, to the worm pellet, add 100 microliters of 40% isopropanol, or 60% for ORO staining, and incubate the sample at room temperature for 3 minutes.
The addition of isopropanol is crucial for proper permeabilization of the worms before staining.
Centrifuge the worms at 560 times gravity for 1 minute and remove the supernatant without disrupting the worm pellet.
It is important to minimize light exposure during the centrifugation steps.
Now, while in the dark, add 600 microliters of previously prepared NR working solution to each sample. Invert the tubes 3 times.